Have you, as Illumina, or as a group of people working in genetics with the same mission, have you seen a sort of standardized data format or set that everyone's running towards, working with, in agreement with? We've seen this across so many industries where formats and data standards have been a huge debate and often a huge blocker in terms of sharing information with one another. Really interested to understand how you have overcome that, especially now that it's very much global practice and it's not just within one country or within one single area.
Yes, it's a great question and a really complicated topic generally. What I will say, even before you get to the data itself, one of the most important, I guess, points to consider is how the individual patient samples were consented. So consent in the proper IRB protocol within the institution. And again, this could be an academic medical center that's linked to a health system or a hospital. It can be just a research lab that works with that has docs that do, it can have all these different flavors, health systems, AMC, academic medical centers, et cetera. Regardless, they have to have, in order to use those precious samples for research purposes, they have to have the proper consent in place. And that consent language and again, within each country, there's some variation, but overall, it needs to be protected. And in US, you have HIPAA, you have GDPR now in Europe. And so the consent language for the samples themselves have to be, that protocol has to be airtight and really done in the proper way. Once you're past that, then you get into, okay, here are the sample types. And again, these biospecimens, you may have whole blood, you may have just DNA that was isolated downstream, you may have tissue if you're talking about oncology indications, et cetera, not just oncology, but primarily that's one of them. So you have these different types of samples. And so the different molecular data you're going to generate from those samples will dictate in kind of what research is being contemplated, will drive the standards and kind of what ends up being implemented. But I will say that from a germline genetics point of view, that 30x coverage, whole genome has really, and I think the standard has kind of been set there for research. At least I wouldn't make that argument. Some might challenge me on that. But if you're looking at large discovery cohorts, getting a 30x whole genome, and you do mapping and alignment, we use the Dragon pipeline for secondary analysis. It's industry leading on the Precision FDA. It is something that we believe and support. You end up with a VCF, which is a variant call format file. And so that is kind of a standard way to interrogate this type of data. And then you look to the phenotypic data that you're going to marry to that. That could be structured clinical data, ideally from the EMR directly de-identified. And so that has to be in a, depending on the consent language that you have, of course, if you have it consented properly, then you can provide that for research purposes. It's fully de-identified. And so that can be used for whatever new breakthrough discoveries that will come. If you're in a European setting, you need to have GDPR compliance in place, and the technology you're using, platforms, cloud, on-prem, regardless, you have to sort of follow these rules and make sure that you're operating as you need to. But essentially, it's still early in the discovery cohort realm. I think I would say that type of data at that depth of coverage, those files are fairly standardized now. I would make that argument. But when you look at other areas, you look at proteomics, you have different platforms that are still out there. Some have been used for the larger population projects. For example, the UK Biobank, they did a subset of 50,000 proteomes of the PPP project, the Pharma Proteomics project that was published in Nature, some of the results from that, which which is really exciting. Linking that to the variants PQTL, which is protein quantitative trait loci. That's an application of a lot of interest within pharma, but that's, again, a new modality. You need to have serum to do that, right? So it really is, I think, based on the resources and the specimens within the institutions, and then what the research goals and the scientific questions that are going to be asked, and sort of linking those together. And then the output from that, of course, has to follow the frameworks within those protocols and make sure everything is, kind of done in a proper way, like I said, around consent language and the like frameworks.